Development of ovule and female gametophyte. Ovule means small egg. In seed plants, the ovule is a structure that gives rise to and contains the female reproductive cells. Ovules, which are born into placenta in the ovary, develop into seeds after fertilization. Let us learn the structure of the ovule and the development of female gametophyte. The ovules have a massive parenchymatous body called the nucellus. It functions as a megasporangium. The ovule is protected or enclosed by one or two protective sheaths known as integuments. The ovule with a single integument is known as unitegmic, and an ovule with two integuments is known as bitegmic. The integuments completely cover the nucellus, leaving only a small opening at one end called micropyle. The ovule is attached to the placenta by a stalk known as funicle, and the point of attachment of the body of the ovule to the funicle is called hilum. The basal part of the ovule, where the nucellus, integument and funicle merge, is known as chalaza. Now let us understand how the ovule and its structure develops. The ovule arises on the placenta as a hemispherical protuberance, which grows into a projecting mass of tissue, the nucellus. The inner integument, which is usually formed first, initiates from the epidermal layer, and the outer integument from the subepidermal layer. The growth of the integument is slower than that of the inner integument. The integument soon covers the nucellus, leaving a small opening at the tip, which is nothing but the micropyle. Development of the megaspore within the ovule or megasporangium is known as Microsporogenesis. The megaspore mother cell undergoes meiosis and forms 4 haploid megaspores. Out of the 4 megaspores, only 1 is functional and the remaining 3 degenerate. The functional megaspore is the mother cell of the female gametophyte. It grows in size and forms an embryo sac. The haploid nucleus of the megaspore divides mitotically, usually forming around 8 nuclei which organize in a definite manner within the embryo sac. 3 nuclei at the micropylar end organize into an egg apparatus. 3 at the chalazial end form antipodal cells, and the remaining 2, called polar nuclei, migrate at the center of the embryo sac and fuse to form a single diploid secondary nucleus. The central large cell of the egg apparatus is the female gamete, which is partially surrounded by 2 lateral synergid cells. The mature gametophyte embryo sac thus has 2 polar nuclei, 3 antipodal cells, the central large cell, that is, a female gamete, and 2 synergids. Since this type of embryo sac develops from a single megaspore and has 8 nuclei, it is said to be monosporic, 8-nucleate embryo sac, or polygonum type of embryo sac. It is the most common type of embryo sac, and it is found in more than 81% of flowering plants. To conclude, we can say that the development of ovule and female gametophyte forms a crucial stage in the process of sexual reproduction in angiosperms.